Hello everyone, we are going to start a new series of videos for science and technology from current affairs for the upcoming prelims examination. We'll take up a topic, discuss it comprehensively, we'll also do questions on them, and we'll also go back in time to cover other important topics of science and tech so that you are comprehensively prepared for science and tech section for the upcoming prelims. A good place to start would be the announcement made by the Government of India in the budget. The budget had various important announcements related to science and technology, starting from the Biopharma SHAKTI program, where 10,000 crore allocation has been done that will be utilized over the period of 5 years. This will be the manufacturing capability that India will enhance in biotechnology. We'll talk about it. Announcement also has been done for India Semiconductor Mission 2.0, Carbon Capture, Utilization and Storage, CCUS. You can take this in environment, but we will be covering it in science and tech portion here as well. Research, Development and Innovation, RDI Fund, and we'll talk about the status of the research overall in science and technology in India. The budget also has announced a fund for the upgrade of astronomy facilities. And after Aditya L1, astronomies, they are very, very important for the coming prelims. We'll also talk about Anusandhan National Research Foundation. Otherwise, if there is no institutional strength, then the fund for the innovation will be meaningless. So there are a lot of things to be talked about, and gradually in bits and pieces, every day we will come up with a video, and subsequently in 3-4 videos we'll finish all of them. Today, we'll talk about the first thing, that is Biopharma SHAKTI mission. SHAKTI here stands for Strengthening Healthcare and Allied Knowledge Technologies in India, SHAKTI. Biopharma SHAKTI is about manufacturing. There are 3 stages if you'll see in the ecosystem of biotechnology. One is policy, and to design the innovative policy. This is the overarching strategy of Government of India, Bio E3 policy. This is the umbrella. This policies, they're at the top. This is the umbrella under which everything that we talk about biotechnology will be talked about. Bio E3 policy is biotechnology for economy, environment, and employment. Came in 2024. So it replaced the previous policies. So at the top, at the apex level, we have Bio E3 policy. Within this policy, there is a layer of research, that is Bio Ride scheme. This layer is for research. And so far, we had National Biopharma Mission, which was the 3rd tier, manufacturing. But this National Biopharma Mission has been succeeded, replaced, and also scaled up by the Biopharma SHAKTI mission. So Biopharma SHAKTI mission will be for manufacturing scale up. And as we see the detail, it will be evidently more clear to you that it is about manufacturing and scaling up the manufacturing. The objective of Biopharma SHAKTI is to develop India as a global manufacturing hub for biopharma. But there are 2 specific focus, and you have to know this. I have a feeling that in the upcoming prelims exam, they would ask you something on biosimilar or biologics. There are 2 terms mentioned in the objective of Biopharma SHAKTI program. A development of the manufacturing prowess in biologics and also in biosimilars. So fundamentally in this video, for much of the time, we'll be discussing these 2 things, biologics and biosimilars. Biologics are complex medicine derived from living organisms, living cells, bacteria, animals' body, and then separate it out, filter it, and inject in human body. So it is developed in a living cell, a bacteria, also fungi, also the cell of other animals, but it is extracted from there and put in the human body. They are large proteins, they're complex, they're not simple molecules like that of paracetamol, like the other chemical drugs. They are biological drugs, sometimes they are living drugs, like we'll talk about the CAR-T therapy, they are living drugs or derived from the living cells. They are not produced chemically in the lab. That is biologics. This logic here is not from maths. Biology, biological, that biologic. So developed from the biological entities, so biologics. And biosimilars are similar to biologics. When you develop a biologic for the first time, you have to go through intensive research, development, and then testing. So you have to see whether it is suitable to be put in human body, or the immune system will fight it out. An autoimmune disorder will be triggered because of injecting something from outside. So it is very delicate thing. Manufacturing is delicate because it is manufactured from the cells. And cells, they are sensitive to temperature, pressure, and other environmental factors. If you do not control that, then it will not be manufactured properly, or something else may be manufactured. And they are complex proteins. You also have to protect the structure of that. So you have to maintain the temperature while management of supply and chain. So dealing with it is difficult, very difficult, but they are a very important section of medicine. As we will discuss in a short while, you'll understand. Biosimilars are development which is targeting not the therapies or testing on the human body. The development of biosimilar is targeting developing something closer to biologics. So biologics are original research, original manufacturing, and everything. Once the patent is gone for biologics, it lapses. Then it becomes an open ground for anybody to produce a thing like that. And you know it has been produced, and you know it has been efficacious. So now the whole goal is to develop something very similar to biologics. And testing is not very rigorous. The testing of biosimilar is to see whether it is really similar to biologics or not. So the regulation and testing is easy since original research work is not involved, patent is not filed, so it is also cheaper. So biosimilars is something that mimics the biologics. Highly similar but not identical version of biological drugs. And these are critical. These are critical for genetic disorders, for autoimmune diseases, for cancers. They're required for development of vaccines. So you know, there are two areas in which the government is focusing, and globally there also a focus. One is the semiconductor chips, those tiny brains of any processor, any electronic equipment. We have to develop that, and the manufacturing of biologics. The two are very frontier thing, high-end technological thing, and very important both for prelims and mains. The goal that the government has set up, as announced in the budget document for the Biopharma SHAKTI mission, is to catalyze the investment in advanced biomanufacturing. And why it is advanced, very soon, in 10 minutes, you'll realize it is not easy thing to deal with. It is really advanced biomanufacturing, and the infrastructure will also be advanced. To reduce the import dependence, we have very high import dependence. India is called as the capital of generic medicine. Indeed it is. But the thing is, we do manufacture 60% of the generic medicine. We manufacture 25% of the vaccines. And when it comes to generic vaccine, it shoots to 60% again. But the thing is that the crucial elements in those vaccines and medicines, the APIs, they are still imported. Manufacturing is done by us, but the crucial things, they are still imported. So manufacturing that crucial thing, like for example, in electronic industry also, a lot of equipments are assembled here. India is a big market, and TVs and fridges and ACs and laptops, so many electronic equipments, telephones, mobile phones, they are sold, but they're assembled here. We have assembly unit. The microprocessors, they are not manufactured here. The crucial things are not here. The rest of the things are also assembled, and some parts are manufactured. So the thing is that we are leading in vaccine development, in generic medicine development, but the crucial part of it, that's not here. And so we become prone to any geopolitical risk, any disruption in supply and chain of those crucial things. Our target, as the government has set, is to capture 5% of the global pharmaceutical market share. So you can see that we are lagging already in this area. The overall biopharmaceutical market is a big market. And we excel in the generic medicines, but the high-end manufacturing is something that has been Achilles' heels across, whether in semiconductor or that is in the core manufacturing. Manufacturing has been Achilles' heel of Indian economy. So we have to work on that. The disease burden of non-communicable diseases, that's also increasing rapidly. And as we'll see in a very short while, that biologics, they are used in non-communicable diseases treatment as well. For cancer treatment, they're used as vaccine. They're used to fight against autoimmune diseases. So it is important for a country like India to have such a target. Previously, we had National Biopharma Mission. This will be now replaced by Biopharma SHAKTI Mission. National Biopharma Mission, first of all, was co-founded by World Bank. But this is completely indigenous, completely on domestic budget. And this was a research support system. Other institutions doing researches, and the support system was provided. This was not an end-to-end manufacturing thing. They had focus on vaccines and biotherapeutics, but this was an R&D early innovation pipeline. So this was a support system. They will do researches and disseminate it to the other institutions to carry forward from there. That was the main crux, the idea of Biopharma Mission. But the Biopharma SHAKTI program, this is an end-to-end ecosystem, and the manufacturing, the trial, the regulation, everything is integrated in the Biopharma SHAKTI program. And Central Drug Standard, this organization that regulates, give license for the drugs, this budget has talked about of increasing its capability so that the quick regulation can be done. As you will understand, India was among the first country to, I'll have the detail here, I'll give you those information, but India was among the first country to start giving license for biosimilars. And more than 130 biosimilars license has been given in India. So when we talk of more biosimilars and biologics, biologics must be properly scrutinized. Otherwise, the immune system will start to fight it. So unless you do the regulations properly, the success of biologics will not be there. It will be a very risky thing. But there was no central component of regulation under Biopharma Mission because it was not an end-to-end thing. So this is more comprehensive. This is more specialized. This finds more niche areas of development of biologics, not just biosimilars. The original research work, not just mimicking the biologics. So this is in a sense more advanced and more comprehensive than National Biopharma Mission. Now there are two terms from the perspective of science, from the perspective of prelims as well, that you have to be very clear with. One is biologics and the other is biosimilars. Biologics are medicines derived from living organisms. Living organisms like bacteria, like the cells of other animals. So the question is, how do you do it? How do you get something from another cell, or suppose a bacteria, what you want? One of the early example of biologics is development of insulin. Insulin required by human body. Insulin is required in digestion of sugars. And if pancreas produces insulin, if it does not produce insulin, then the sugar in the blood will not be digested. So then it will accumulate and then it will create problem. The blood pressure will increase, and the blood that is in touch with other organs, the pressure on them will increase and the organ failure will begin. It's not a good thing. The sugar remains in the blood. It needs to be digested, filtered out, but it is not happening. Why? Because insulin is not there. So we have to get it from where? The ingenious idea came to get the insulin from bacteria, because bacteria also produces something similar. Because bacteria also digest its food through the same process. That's why you have a rotten tooth. The tooth enamel, the tough layer, the tooth enamel gets corroded. If you eat something sweet, if you eat something, any carbohydrate you eat and you do not do brush, then the bacteria comes to eat the food, and bacteria will release those enzymes to digest it, and that dissolves the tough upper layer of teeth, the tooth enamel. So the thing is that bacteria do produce such a thing. And through the recombinant DNA technology, we can re-engineer the bacteria to produce something of our interest. The recombinant DNA technology, as the name suggests, that you will recombine. You will recombine the DNA of the bacteria with something that you have. So I will tell you very simplistically at another time and another occasion, we will discuss recombinant DNA technology full throttle. But right now, the topic we are dealing with is biologics. And biologics means that you develop medicine out of living organisms. You can develop proteins, antibodies, nucleic acids. They are large and complex, unlike the chemicals used in the synthetic medicines. So they are not like paracetamol. They, like insulin, will be biologics. They are complex, large medicines, large structures. Like a way of thinking could be that the conventional drugs like paracetamol is like a brick, simple, small, and can be scaled. And identical copies can be made. Very easy to deal with brick. But a biologic like insulin is like a cathedral. It's enormous, complex, made by living cells, incredibly difficult to replicate exactly. So even when the companies are developing biologics, every batch will not be same. There's slight difference in every batch. So copying it in toto exact totally is very difficult. So biologics are not simple molecules, and they are not made in the labs synthetically. Although they'll be made in the lab, but there'll be a specialized setup in the lab, and use of living cells will be there. Now I'm going to give you a little bit. You would already know recombinant DNA technology, a very important topic. I'll discuss all the biotechnology related technologies like recombinant DNA technology, like RNA interference technology, like CRISPR-Cas9, like monoclonal antibodies, etc. In one go in some other videos. But right now, let me give you an idea of it. And you already know it. Let's discuss it a little bit. Recombinant, as the name suggests, you'll recombine. Suppose you take a bacterial DNA. In a bacteria, the DNA is not inside the nucleus. It's there in the cytoplasm in a circular form like this. So what you have to do is you have to insert a part of human DNA into the bacterial DNA. So you put that in the vector gets inside the bacteria, releases it in the cytoplasm of bacteria. And the mechanism of the bacteria itself inserts it inside the DNA of the bacteria. It does it by itself. It does it by itself. It is not like CRISPR-Cas9 that you go to the right point and do the cut and pasting. No. It's not that specialized. It is a very old technology, and this insertion can happen anywhere in the bacterial DNA. It's not a precise cutting. It's not a precise insertion. It can get inserted anywhere. And when it gets inserted, then the normal activity that happens to DNA, whatever it is coded for, happens. But a new code has been added. So this code will also run in the machine. Meaning this code will also be read by the ribosome of the bacteria. And ribosome will create something which doesn't belong to it. But nevertheless, code has been added. So it will start to produce thing corresponding to this code, depending on what the code is. So if you code it, if you insert the code of the human insulin, whatever the code is to produce human insulin in human body that is there in the DNA, this bacteria is also going to produce it, because ribosome can create it because it already creates something similar for the bacteria. It will create something not required by bacteria, but will be there in the cytoplasm of bacteria, which can further be extracted. This is biologics. Getting your medicine by a living cell, this is biologics. And this is not easy, obviously. It's a complex thing. It's a very complex manufacturing where you're using the tools and you're using the living cells as your machines. The living cell produce something and give it to you. It's a ribosome of the bacteria which is producing it and giving it to you. They are the machines. But to work out with these machines are not easy. But they are very useful. Very, very useful. Very useful. So this is how it's done. Recombinant DNA, you insert it into the host cell, and these living cells become the factories, because the factory will produce whatever the coding is in the manufacturing unit, and you added a code. They will not question it. Whatever code has been given in the machine, the production line will not question it. It will just accept it because it has been accepted by the DNA. DNA is the boss. Whatever is in the DNA, everything happens according to it. So if you can successfully manipulate the DNA, rest of the body will not question it. It will simply do whatever the commandment is. You have added a new commandment, that will also be followed. So it will produce unnecessarily. It doesn't require it. But since the code is there, it will produce. It will not use, just produce. Who will use? We will use. We will separate them out, extracted, purified, and formulated into medicine. But since these are living cell factories, the tiny variation in temperature, pH, or nutrients, or culture condition, any condition, can change the final product. So this is what makes biologics so difficult to copy perfectly. That's why when you're trying to reach here, you end up in biosimilars. Can never produce the exact copy. Even the manufacturers of biologics, their different batches differ slightly. This slide has the detail of recombinant DNA technology, but this is not the topic for today, and some other day I'll do it, but this PPT will be attached in the description of the video, so out of interest you can read it. Biologics can be of many types. Certain things are important and you have to know for your prelims exam. Monoclonal antibodies. Monoclonal meaning the same kind of clone without any variation. Just single copy will be multiplied, that is monoclonal. Antibodies, you understand, antibodies are the real thing that kills the antigens in the body. We survive because of antibodies. When a pathogen enters inside human body, bacteria, fungus, anything, they are killed, they are neutralized by the antibodies. Antibodies are the most potent defense mechanism in our body. But if a person is immunocompromised because of old age, because of comorbidity, the immune system is not working, that person will not survive infection. Or the immune system working very slow. It will take 22 days to produce antibodies. In normal, healthy adult, it takes around 3 to 7 days to develop the antibodies. So if you get cough and cold, you will be having the cold for some time, 3, 4, 5, 6 days maybe. But eventually you will get better because antibodies has been made and the viral infection has been killed. The virus that entered that is responsible for the cold, that has been neutralized. So you will get better. It will take time. But in old aged people or immunocompromised people, this time duration is too long. And by that time, the virus or the bacteria will completely overwhelm the body. And anybody can potentially die. So what the idea is that antibodies have to be injected because immune system is not producing antibodies. The antibodies have to be injected from the plasma of some other person. So if a person got healthy from the same disease like COVID-19, a person got healthy, got infected, got tested, the test was positive, and now the test is negative. Now he is healthy back again. That means 100% he is carrying the antibody because without antibody, he will not get healthy. So you can extract the antibodies from the person, and then inject into a cancerous cell. A thing about cancerous cell is that they multiply like crazy. Super fast speed they multiply. Some of them are also called as immortal cells. They never die. They keep multiplying. So you inject the antibody of the person who has got healthy, antibody responsible for that particular disease into the cancerous cell multiply. And when the cell multiplies, along with it, the antibody also multiplies. Because whatever is inside the cell, this nucleus and ribosome and lysosome and endoplasmic reticulum, the vacuoles, whatever, everything multiplies into the daughter cell. In the daughter cell, you will have the full copy. If you have something injected, like the antibody injected, that antibody also multiplies. If you have viral infection in the parent cell, daughter cells will also have the viral infection. If you have the RNA of virus somehow in the cytoplasm, this you may have never thought about it like this, but this RNA will go in the daughter cell as well. Otherwise, the disease will not persist. There's a lifetime for the cells. If the extra thing does not pass on, then the infection will not pass on. So the daughter cells will still be healthy, and this cell will die after some time. So you will get better, anybody will get better of any disease eventually. But it doesn't happen like that. The daughter cell also carry the infection. Meaning whatever is in cytoplasm, everything is made a copy of. If you put the antibody here, that will also be made copy of. And that multiplies and multiplies and multiplies. So the antibody from one human body, one healthy person, cannot be given to plenty of other patients already in the hospital. But if you multiply, then you have too many, and from there you can distribute. So you can manufacture the drug from the antibody of one person through this. So that is monoclonal antibodies. That is the mechanism. I'll discuss all these techniques again in a separate video. Monoclonal antibodies and RNA interference and CRISPR-Cas9 and stem cell therapy, all the major biotechnology related technologies I will discuss with you. Right now, I am here only to tell you a little bit of biologics. That's it. So this is one kind of biologics. Anything that is developed from the biological system, living cells, that will be referred to as biologics. And last large section of drugs that is from this category, biologics. A new emerging drug is CAR-T, chimeric antigen receptor T cell, CAR T cell therapy. Chimeric related to cancer, antigen is anything which is on infection or a foreign body. I hope you have a little bit of idea of cancer. In cancer, what happens is that the cell get misprogrammed and looks different and doesn't perform the normal function, and it multiplies like crazy, difficult to control it. So the cancer looks different, and you train your T cell to recognize those cancerous cells better and kill them. T cells are an important part of immune system. T cells are killer cells. So anything outside, even cancer cells, are killed by T cells. But the T cells, imagine like T cells are sent to the police training center, and they're trained very well to identify the criminals and come back. So T cells comes back into the body. You extract the T cell of the person and you engineer the T cells, and then you put back into the human body. So these transformed T cells, engineered T cells, are very well suited to fit into the cancerous cells, latch on it, get hold of it, and neutralize it. You know how does a T cell kill? Or how does antibody kill the antigens? By latching onto it. Because in the blood, everything is rolling. Everything is rolling. Difficult. Anything cannot kill anything because everything is rolling. Imagine two people are skydiving. Difficult to fight in the air. Unless you get hold on the of the person, you cannot neutralize the person in the air when both of you are falling. Similarly, in the blood, anything to kill anything have to get hold of something. So latching is very important. Without latching, neutralization cannot be done. So the T cells, they are reprogrammed to develop something on their surface that latches very well with the particular cancerous cell. And then it becomes effective in neutralizing the cancerous cell. So it's also a drug. It will also come in the category of biologics because you are enhancing the efficacy of T cell. So you are dealing with a living cell. When you deal with living cell, use that as a drug in any ways, that will be in the category of biologics. So this is an important one, and surely I'll discuss with you separately, more elaborately in some of the other topic. But CAR T cell therapy, damn important for the coming prelims. Monoclonal antibodies, last year they have already asked. The recombinant proteins, vaccines, mRNA vaccine, they already have asked. So all three were important, and we knew it would come. This has been asked, and this is yet to be asked. So all of them are in the category of biologics. Biologics can be used to treat cancer, autoimmune diseases, diabetes, genetic disorder, infectious diseases, neurological problem. They all can be treated in one or the different way. Like the cancer cells can be killed better by a re-engineered T cells. The autoimmune disease can be treated. Autoimmune meaning immune cells are attacking us. Immune cell has to attack pathogens, but autoimmune disease is the immune cell is killing the healthy cells. So they need to be suppressed. For that you require suppressants. That is also protein. Those special proteins are made and injected in the human body to suppress the immune cells. Immune cells are suppressed actually. After a while, the immune system are calmed down because the pathogens are killed. But if the body fails to calm them down, they will not calm down. So because the body is not manufacturing those proteins that calms them down, so you have to put inside the human body as a drug. Has to be manufactured again. So these proteins, which are the controllers, they are signaling mechanisms, they are the building blocks, they are everything in the body. So if they are failing, we are failing to manufacture insulin, their enzymes, their hormones, their signaling mechanisms, their structure building blocks, they're everything in the body. If that is not being produced, it has to be produced from outside and injected in the human body. So it is very important, very, very important. Biologics are expensive, as I explained you, difficult to deal with the living system. Manufacturing something from the living system and this lower temperature in transportation and etc. This is an expensive, very, very expensive affair. Biosimilars, biologics, when the patent of the biologics expire, then it becomes open for anybody to manufacture something like that. Then the manufacturers can develop a version that is highly similar but not identical. Actually, it is not restricted by law, but you will not be able to do it by the living cells mechanisms. Whatever you produce will not be exact same as original, but will be quite similar. Since it will be quite similar, so it will also be similar in its efficacy. That is called as biosimilar. A biosimilar is to biologic what a generic medicine is to conventional drug. But here is a catch. Generic medicine is exactly same chemically as the conventional drug. And the other supporting chemicals could be different. Chemically, generic medicine is exactly same. But biologically, biosimilar is not exactly same to biologic. It is similar. Name suggests it is similar, but not same. But generic medicine's main chemical is exactly same as the conventional drug. I hope, yeah, there's a critical difference. A generic medicine is chemically identical to original, while biosimilar is only highly similar. It is not identical. And this difference exists because biologics are made by living cells, and no two batches from even the same manufacturer are perfectly identical. I hope now things are clear to you. Biosimilars, they are very important, and recently, it is in why news and government has planned, because a lot of patent cliff opportunity is going to come between 2025 to 2032. There are many biologics whose patent will lapse. They are losing the patent protection, and India already has a base in generic medicine. Generic medicine is different. I hope you understand. That is a chemical. Biologics, they are not chemicals that you deal with. You deal with living cells, and living cells are your machines. So the generic medicine ecosystem we have and the expertise we have, but a new set of expertise has to be developed afresh, anew. But advancement there obviously will help. The human resource is there, and the same human resource and the same laboratories can be used, which were used for generic medicines can also be used for biologics. But excelling in generic medicine by itself does not mean that we will excel in biologics as well. But we do have an advantage, and we must take this advantage and take the first mover advantage in developing biosimilars for the biologics whose patents are going to expire. India was one of the first country to approve a biosimilar. And India approved its first biosimilar way back in 2000. We have 135 plus approved biosimilars, more than any other country. And companies like Biocon, Dr. Reddy, Cipla, they have done really well in developing biosimilars. So it has a huge export opportunity, and the rising burden of non-communicable diseases, it can also be dealt very well using the biosimilars. So it has to be done. It has to be done. That is why it's important. By this time, I hope the difference between biologic and biosimilar is clear to you. It's a medicine from living organism, and it is also a medicine from living organism, but it's a near copy of the biologic after the patent has expired. Its original R&D is required for this. R&D will also be required for this, but not to that extent, because it is not an original thing. It's just a follow-up to the biologic. The cost will be very high. It will be cheaper, much cheaper than the biologics. And full clinical trials are done for giving license to the biologics. Biosimilars licensing is not on clinical trials. It is on the basis that how compatible or comparable it is to the biologic. If you can prove that your protein is comparable to biologic, then you get the license. Clinical trials are not required for licensing of biosimilars. So why do we focus on, why are we focusing on Biopharma SHAKTI? Specifically, we are focusing on biologics and biosimilars rather than conventional pharmaceuticals. I hope you know the difference now. Conventional pharmaceuticals will not be effective in the autoimmune diseases or the complex protein that requires to suppress the immune system, the protein which is required to make the vaccines, the protein that will be required for growth hormones, insulin. You cannot do that synthetically in the lab. Very difficult. So it is something that has to be focused on, and I've given the reason why we are focusing on the biologics. That part will be important for your mains exam. Now, whatever we have studied, well, let's do the conceptual assessment as to how far you have understood them. Biopharma SHAKTI is the first government initiative in India to support biopharma manufacturing. Biopharma manufacturing was also supported by National Biopharma Mission. So it is not the first one. This is plainly false statement. A biosimilar, once approved, can be considered chemically identical to its reference biologic. It is never similar. It's never identical. Biosimilar is a close similar. It is never identical. So it's a false statement. You must think of true and false ahead of me before I declare it. You must get your answer. The difficulty in manufacturing biologics arises primarily from the high cost of raw chemical inputs. The difficulty in manufacturing of biologics arises not primarily from the high cost of raw chemical inputs. Raw chemical inputs are not used here. Primarily because you're dealing with living cells and manufacturing from living cells, the environment has to be stabilized. That is a prime difficulty. India was late entrant in biosimilar. India was one of the first country to give license for biosimilar, right? Biopharma SHAKTI will be jointly administered by Department of Biotechnology and Department of Pharmaceuticals. This information was there in the first slide. I did not read it, but this is true and this is important for your exam. Monoclonal antibodies are classified as biologics because they are produced by living cell system. Correct. That's why they're classified as biologics. Cell and gene therapies like CAR T and CRISPR based therapies are classified as next generation biologics. That is right. In CAR T, I explained you deal with T cells. You deal with T cells, so you are dealing with living cells. And in CRISPR-Cas9, you do some engineering on the living cells to convert it into maybe a drug or to make it better. So this is right. That this will also be counted under the biologics. And India already has started to focus on CRISPR. In some other videos, I'll give you the detail on CRISPR and other technologies of biotechnology. Just stay tuned. I'm going to dole out everything important for the coming prelims in this series. India's historical success in generic conventional drug manufacturing is a sufficient foundation to lead in biologics. Conventional generic drugs, they are chemicals. They're not biological. So it will not be sufficient to advance in biologics. You understand that. This is a false statement. Let's do this question. It is regarding Biopharma SHAKTI. Consider these statements. It is administered solely by Department of Biotechnology. False. Jointly by Department of Biotechnology and Department of Pharmaceuticals. It is explicitly linked to India's Bio-E3. Everything will be linked to Bio-E3 policy. That's the APEX policy. It has to be true. It is primarily focused on biologics and biosimilars. Exactly. This is the whole point. That's why we had so much of discussion. This is true. So answer should be B. Now the last question, consider the following statements regarding biosimilars. A biosimilar must demonstrate chemical identity with the reference biologic to receive regulatory approval. Chemical identity. Well, that is tricky because it is similar, not identical. And I think this will make the statement wrong. Biosimilars can reduce treatment costs. That is right. India approved its first biosimilar before United States and European Union. This is also right. Very important statement. You have to remember this. So again, the answer will be B. This will be the way we will discuss every day. Topic, their concepts. We'll do more than what is presented in the news, more than what's there in the budget document. Comprehensively, I'll prepare you for the prelims exam. Things that goes around it, we'll discuss that as well. We'll discuss questions, how questions can be framed, what statement can be there. In the past, how UPSC has done it. Like the departments, they give it wrong in the previous question. That is something UPSC does focus upon. So based upon those experience, I'll give you the MCQs as well and some basic true false statement as well. That will be fun. So see you again tomorrow.